The animal known as the saber-toothed tiger never existed. The name itself is the first thing the DNA dismantles. Smilodon fatalis, the species that has appeared in animated films to natural history museums, always rendered as a lion-shaped predator with oversized fangs and an implied kinship with modern big cats, is not a tiger. It is not closely related to tigers. It is not a close relative of lions, leopards, or any living cat species in the sense that popular culture has consistently implied. When researchers finally extracted and sequenced its genetic material, what they found confirmed a phylogenetic distance so profound that the entire framework most people carry for this animal had to be rebuilt from the ground up. The DNA placed Smilodon in a completely separate evolutionary branch that diverged from the ancestors of all living cats approximately 20 million years ago, before the ancestors of lions, tigers, and domestic cats had even differentiated from one another. Begin with why DNA recovery was possible at all. Smilodon presents a situation that is essentially the mirror image of Tyrannosaurus rex. T-Rex is 66 million years old, and at that age, the DNA is gone, degraded beyond recovery, replaced by mineral, detectable only in trace protein fragments. Smilodon fatalis, by contrast, went extinct roughly 10,000 years ago. At that timescale, DNA preservation becomes genuinely possible, particularly in cold or dry environments where degradation slows. The Brea tar pits in Los Angeles have produced thousands of Smilodon specimens, more than 2,000 individuals, preserved in asphalt that sealed them from oxygen and moisture. Some specimens recovered from permafrost-adjacent sites in South America retained bone collagen intact enough for extraction. In 2017, an international research team published the first successful retrieval of mitochondrial genome sequences from ancient saber-toothed cat specimens. They had managed to read the genetic text of an animal that had been extinct for 10 millennia. The question was what that text said. What it said was not what anyone expected. The 2017 study, published in Current Biology by Pymans and colleagues, reconstructed mitochondrial genomes from two separate lineages of saber-toothed cats, Smilodon, the iconic American genus, and Homotherium, a scimitar-toothed relative from Europe and North America. The researchers expected to find two closely related species. After all, both were saber-toothed, both were large felid predators, both had evolved the same dramatic dental weaponry. What the mitochondrial sequences showed instead was a genetic distance between Smilodon and Homotherium that was greater than the genetic distance between a tiger and a house cat. Two animals that look strikingly similar, that share the defining characteristic of enlarged upper canines, that occupied similar ecological niches, that have been grouped together in museum exhibits and popular illustrations for over a century, turned out to be more distantly related to each other than species we treat as almost comically different. The DNA had measured the resemblance and found that it was almost entirely superficial. Then, the Homotherium data produced a finding that rewrote a different part of the timeline. Among the specimens analyzed in the 2017 study was a mandible recovered from the floor of the North Sea, dredged up by fishermen and eventually reaching the hands of researchers who recognized it as Homotherium. The established timeline for Homotherium in Europe placed their extinction at roughly 300,000 years ago. The North Sea specimen was 28,000 years old. DNA confirmed the identification beyond doubt. Homotherium had not disappeared from Europe 300,000 years before the present. It had been alive, in Europe, 270,000 years later than the fossil record had suggested. And it had been alive at the same time as modern humans. The DNA did not merely clarify a relationship. It collapsed a timeline that paleontologists had built over decades and replaced it with something no one had predicted. The nuclear genome came next, and this is where the picture became genuinely strange. A draft nuclear genome of Smilodon populator, recovered from a specimen dated to approximately 13,000 years before present and published via the National Science Foundation Paleogenomics Initiative, gave researchers the first look at the full genetic architecture of a saber-toothed cat. The analysis confirmed what the mitochondrial data had suggested. There was no gene flow between Smilodon and any living cat species. The divergence was clean, ancient, and complete. But the nuclear genome also allowed researchers to begin asking questions that mitochondrial DNA alone cannot answer. Questions about gene function, about which biological systems were under selection, about what the genome of a saber-toothed cat was actually doing that modern cat genomes are not. What the genomic data revealed about the fangs was perhaps the most conceptually disorienting finding of all. The saber-tooth trait, the dramatically elongated upper canines that define the popular image of Smilodon, did not evolve once. The genetic analysis, combined with the fossil record, confirmed what paleontologists had long suspected but could not fully establish with bones alone. The saber-tooth form evolved independently from different starting points in multiple entirely separate lineages across millions of years of mammalian evolution. Thylacosmilus atrox, a predator from South America, was not a cat at all. It was a sparacidont, a distant marsupial relative, and its saber teeth evolved on a completely different genetic and anatomical foundation than Smilodon's. Nimravids, sometimes called false saber-tooths, developed the same elongated canines independently in another separate lineage. Even within the Machaerodontine branch that includes Smilodon and Homotherium, the specific mechanics of the canine hypertrophy appear to have been solved differently in different genera. The fangs were not an invention. They were a solution, one that evolution found over and over, by different routes, whenever a large predator occupied a niche that required a method of killing large prey that modern cats cannot replicate. Which brings the question of behavior into direct conflict with the most familiar assumptions. The biology of the fangs has been debated since the first Smilodon skull was described in 1842. The canines of Smilodon fetalis reached approximately 17 to 20 centimeters in length, were laterally compressed, and were serrated along the front and rear edges. They were not, as early reconstructions sometimes suggested, for stabbing prey through thick hide and a direct forward thrust. Biomechanical analysis of skull geometry indicates that Smilodon's jaw opened to an angle of approximately 120 degrees, far wider than any modern large cat, and that the killing method likely involved a precise downward bite to the throat or back of the skull of immobilized prey. The canines functioned as precision instruments, not battering rams. They required the prey to already be controlled, which in turn raises the question of how animals weighing 160 to 280 kilograms were immobilized in the first place. The answer the fossil record increasingly supports is that Smilodon did not hunt alone. Evidence for social behavior in Smilodon comes from multiple independent lines of data. Healed injuries found across Brea specimens, broken ribs, fractured vertebrae, severe limb damage that shows clear bone remodeling indicating survival over months or years, suggest that injured individuals continued to receive food from somewhere. A predator with a fracture severe enough to prevent hunting could not have sustained itself on scavenging alone in competition with other megafauna predators. Survival required provision. A published study documented a Smilodon individual that had developed hip dysplasia severe enough to prevent normal locomotion from a young age, and had nevertheless survived to adulthood. The logical inference is group provisioning, something structurally analogous to the behavior seen in modern lions, where incapacitated group members continue to access kills made by others. The DNA cannot directly confirm social behavior, but it can place Smilodon within a phylogenetic context where the behavioral flexibility required for sociality was already present in related lineages, and the fossil record fills in the rest. The extinction, when it came, was rapid and complete.
Smilodon disappeared approximately 10,000 years ago at the end of the Pleistocene as part of a wave of megafauna extinctions that swept through the Americas following human arrival. The genomic data adds a dimension to this story that bones alone could not provide. Smilodon's killing apparatus was highly specialized, optimized for large, slow prey that had never encountered an ambush predator with tools. The megafauna that Smilodon had co-evolved with over hundreds of thousands of years, ground sloths, mastodons, glyptodonts, giant horses, were disappearing simultaneously under hunting pressure from a new kind of predator. Smilodon's saber teeth were precision instruments, tuned to prey that was vanishing. The 2021 nuclear genome analysis found no evidence of genetic diversity loss in the period immediately before extinction, which suggests the collapse was abrupt rather than gradual. A population crash compressed into perhaps a few thousand years, rather than a slow fade driven by long-term inbreeding or climate pressure. The animal did not decline. It fell. What the DNA leaves behind is a portrait of something almost nothing in the popular image prepared for.
Not a tiger, not a relative of lions, an independent evolutionary lineage that separated from the ancestors of all living cats before those ancestors had differentiated into the species we recognize today. An animal whose most iconic feature, the fangs, had been invented and reinvented by entirely unrelated lineages across 50 million years of mammalian evolution, because the solution those fangs represented was simply that effective. An animal that may have been social in a way that no popular reconstruction has ever depicted. An animal that survived in Europe until modern humans were carving figurines and burying their dead, until a North Sea mandible preserved in sediment for 28,000 years told researchers that the extinction date they had written down was wrong by 250,000 years. The name is wrong. The image is wrong. The family tree is wrong. The timeline is wrong. The DNA corrected all of it, and what it replaced those errors with is considerably more interesting than anything the original story contained. Subscribe. New discoveries about lost species posted every few days.